about contemporary evolution, and I think what all the other talks so far have set up, is that where we expect to see a lot of contemporary evolution is in response to invasive species, of invasive species. So you're taking a species, you're putting it in a completely novel environment, and so there's a potential to be a lot of selection pressure there. And so a lot of that selection pressure may come either during the invasion process or shortly thereafter. So if you have some pool of invaders coming into a community, there's potentially biotic resistance to that invasion, provided by a number of different species. So in a really simple example of that, as bullfrog larvae try to invade ponds, they're eaten by dragonfly larvae. And so in the presence of dragonflies, you see no survival of those bullfrogs, but actually significant survival in the absence of them. And that biotic resistance may be provided by a lot of different things. So it might be from predation, parasitism, herbivory, competition. And we usually think about these negative species interactions driving that. And we rarely think about mutualisms, particularly the lack of mutualists driving biotic resistance. And so Zoe Lopez gave a really nice talk this morning showing that for legumes trying to invade communities, a lack of rhizobia in those communities might provide some resistance to invasion. So if we think about that pool of invaders coming in, that biotic resistance might be variable in who it's resistant to. And so maybe some genotypes can pass through that biotic resistance, and that's effectively selection, right? So some genotypes make it through, they're the ones that survive and reproduce into the next generation. And so that biotic resistance might provide some selection on those invasive species. And that's where we would see this rapid contemporary evolution. But of course, species in communities are really diverse. So they're rarely interacting with one other species, they're interacting with many other species at the same time, and there could be some community context to that evolution. And so if we're trying to put together this whole puzzle of the community, we have to consider lots of different puzzle pieces simultaneously. And so when you have lots of species there, there's potential for indirect effects to occur. So if we stick with the same example of the bullfrog and the dragonfly larvae, we see this result if just those two species are present. But if we add fish into that community, the resistance provided by dragonflies is significantly less. Now some of those bullfrogs do make it through. And so those indirect effects alter the effectiveness of that biotic resistance, but they might change the evolution imposed by that biotic resistance too. So it might be that if we add indirect effects into communities, instead of just some genotypes coming through, we might have more genotypes coming through. Or we could have the opposite. Those indirect effects might make the biotic resistance more effective rather than less effective. And so those have the potential to alter the selection pressure on that invasive species. So I'm going to talk about some work I started in Jen Lau's lab, the postdoc at Michigan State, and it's continued since then. It's been done by several different people over time, lots of different experiments. So I'm going to try to kind of sum up about 7 years of experiments in about 5 minutes. So we're looking at Medicago polymorpha. So this is burr clover. It's a common invader around the world. It's introduced in most places probably as a forage or a cover crop, and then has escaped into natural communities. It's invaded lots of different parts of the world, so it's native to sort of the Mediterranean and Western Asia. It's invaded almost every other continent except for Antarctica. And so we've taken genotypes from all of those different locations. This is a selfing species, and so effectively each plant is a different genotype. And so for different experiments, we use different numbers of replicates, but we've usually got between 35 and 40 genotypes. And then those are split between the native and invasive range. And then we do these experiments with several replicates of each of those genotypes. So we can look at the effect of whether things came from the invaded or the invasive range. We can also look at effects of specific genotypes on traits, which I'm not going to talk about much today, but there is a lot of variance there as well. So Medicago, in its invasive range, at least in much of its invasive range, interacts with a lot of these other species. So it experiences herbivory by these alfalfa weevils. It competes with other species. In California, where we do a lot of our field work, it competes with another legume, acnospawn, pretty frequently. It also is a legume, and so interacts with rhizobia bacteria in the soil that form these nodules on the roots, and the plant gets nitrogen from those rhizobia. And then because it grows in these sort of Mediterranean or dry climates, it experiences a lot of water stress too. So I'm going to throw water limitation in here, even though it's not a biotic interaction. It still has the potential to impose selection on this species. So it's interacting with all these different species at the same time, and there's potential for indirect effects to occur there too. So for instance, you could draw these indirect effects in a lot of different ways and through really complex pathways. One, for instance, that we've looked at is that Medicago, when it's growing next to a competitor like Acmospawn, it experiences more herbivory than if it was just growing by itself. Probably because those herbivores are queuing in on the competitor plant, and then they switch over, and Medicago gets more herbivory growing next to those target plants. So what we're going to do is ask whether plants in the invasive range differ from plants in the native range, and so we're going to look at interaction with one species and just look at whether those native range genotypes differ in those interactions from the invasive range genotypes, suggesting that some evolution has occurred either in the native or invasive range in different ways. So our approach is that we grow all these genotypes at least for one generation in a common garden in the greenhouse to get rid of or at least minimize the maternal effects that might have occurred from things stored in the seeds. And then we do all these experiments in a common garden, and so we shouldn't see any plasticity effects. So any difference we see between native range and invasive range genotypes should be due to some genetic difference there. So a lot of this field work was done at the M'Laughlin Reserve in California, but there's also a lot of this that's been done in greenhouses, either in Michigan or in California. And so the experiments are relatively simple, or at least I thought a lot of them would be relatively simple. Turns out when you plant thousands of seedlings out in the field, most of them don't survive. So there are low survival rates on some of these, but we still find significant treatment effects in many cases. So one thing that we've done is manipulate insects in the community. So we just have plots that we spray with insecticide, plots without insecticide, and we have replicate plots with replicates of replicate genotypes in those plots. We've also looked at intraspecific competition. So either we have Medicago growing by itself, like one pot per plant, or Medicago growing 2 plants per pot. And so they're just experiencing competition from usually the same genotype. We've looked at interspecific competition with replacement design. So either we're looking at plants growing with conspecifics or plants growing with some other competitor. We've looked at grass, an invasive grass in California that's a common competitor, and another legume as well. And then we also have looked at mutualists. So we've tried experiments in pots where either we inoculate the pots with rhizobia or just have sterile soil in them, so they have no mutualists there. And then finally, we manipulate water limitations. So either the plants are well watered or they're kept at some low water level to the point where they're at least slightly stressed. And so jumping into the results, if we look at herbivory, what we find is that, I'm going to surely hit the wrong button here. I will hit no buttons. So if we look at some estimate of fitness on the y-axis, and we look at the native range genotypes in green, they're affected by insects. So if you have insects in those plots, fitness goes down. But if you look at invasive range genotypes, fitness goes down even further. And so for the rest of the talk, instead of showing you this sort of plot, I'm going to show you this sort of plot, which is just effect sizes. Right? So we can see the effect of insects on invasive range genotypes is greater than the effect of insects on invasive or native range genotypes. And here, we're eliminating most insects from these plots. We're assuming most of these effects are from herbivory, and that's backed up by estimates of leaf damage. To look at some other interactions, we see that in regards to intraspecific competition, native range genotypes are affected pretty negatively by that competition. Invasive range genotypes not really affected very much at all. And so it might be that invasive range genotypes have some increased competitive ability there, or at least competitive response. We've also looked at interspecific competition, and we don't find very much effect there. So that's sort of surprising that looking at several different competitors, we don't find very much difference between invasive and native range genotypes in their response to interspecific competitors. Looking at Rhizobia mutalis, native range genotypes tend to benefit a lot from adding Rhizobia into those pots. Invasive range genotypes don't really seem to care if there's Rhizobia there or not. If anything, they experience a negative effect of those rhizobia, suggesting they're maybe slightly parasitic. And then with regard to water limitation, native range genotypes are affected quite a bit when they're limited by water. Invasive range genotypes still experience a decrease in fitness, but not nearly as much as the native range genotypes. So just to sum up those direct effects, those invasive range genotypes are more affected by insect herbivory. They're less responsive to interest-specific competition. They're less dependent on rhizobia, and they're affected less by water stress. And we don't know what the ancestral state of most of these populations was, and so we don't know if the invasive range genotypes have evolved to be less dependent on rhizobia, say, or if the native range genotypes have just evolved to be more dependent on it. So I can't really say anything about the direction of evolution, just that it's happening differently in native and invasive range populations. And so, of course, these are just direct effects. But if we look at other species and we add indirect effects in there, those direct effects may be altered by the presence of additional species. So we did a whole other suite of experiments in which we manipulate not just the presence of one species, but the presence and absence of two species in a factorial design. And then we can look at direct and indirect effects. So we've looked at manipulations of competitors and mutualists at the same time, and we've looked at manipulations of mutualists and water limitation at the same time. And what we find is that if we just look at the effect of rhizobia, native range genotypes don't really care much about the presence of rhizobia. Or at least they don't care differentially between when they're in intraspecific competition or interspecific competition. They benefit from rhizobia the same way in both of those competition treatments. Whereas invasive range genotypes, if they're in intraspecific competition, they tend to be hurt by those rhizobia being present, but they benefit from those rhizobia in the presence of interspecific competition. So maybe they're getting maybe that nitrogen now matters once they're in competition with other plant species. Similarly, if we look at the effect of water limitation, for the native range genotypes, they experience a benefit from rhizobia in their response to water limitation. They have less fitness effects from water limitation if rhizobia are present. Invasive range genotypes are just the opposite. They experience more detrimental effects of water stress if rhizobia are present. So these indirect effects are having a big effect on selection on the plant. And so they're responding differently in the presence of those indirect effects than just in response to direct effects of those species. So as everyone else in this session has demonstrated, invasive species experience this novel selection pressure that can drive contemporary evolution. Both direct and indirect effects can drive evolution in a community context. And so if we're trying to put together this puzzle of the community, we need to consider the more puzzle pieces, the better. And if we also consider how those puzzle pieces are changing shape, that's going to affect how the whole puzzle gets put together. And finally, yesterday I joked that a talk is not worth anything unless you can tweet something about it afterwards. So there's the tweet that sort of summarizes the talk. And I might have like 1 minute for questions.